This video demonstrates the use of CaPTk in characterization of breast parenchymal patterns from digital mammograms, focusing primarily on the assessment of breast density. Here you can see the graphical user interface of CaPTk. Mammographic breast density refers to the extent of radiodense tissue as reflected in images. Here we see some examples of digital mammograms presented in order of increasing breast density. Breast density is considered a very important biomarker in breast cancer screening because it is associated with both aspects of breast cancer risk. It is an established risk factor for breast cancer development. Also, it can substantially affect the sensitivity of mammography due to masking of tumors within dense breast. Recognition of breast density impact on the implementation of personalized breast cancer screening has generated increasing national interest, with many states' legislation currently mandating notification of an individual woman's breast density in mammographic screening results letters. Breast density has gained tremendous research attention, with key studies showing not only that breast density can improve risk assessment models, but it can have a shared genetic basis with breast cancer susceptibility, which is associated with biological phenotypes of tumors and a risk factor for benign breast diseases. Our LIBRA application has been designed for automated assessment of the absolute area of dense tissue within the breast, DA, as well as the breast percent density, PD, and it is applicable to digital mammograms of various vendors. LIBRA has been used in several studies for different institutions and for various applications, including the validation of risk prediction performance of LIBRA density measures, the identification of racial disparities in density, as well as the study of expected density changes over time. The following video demonstrates the use of LIBRA through CaPTk.
Load the mammography image to be processed. The user needs to open the Breast Density Estimator LIBRA single image mode underneath the Applications menu. Since this application has been written using MATLAB, the compiler runtime is being called to handle the computation, and the output from the command line with various computation steps is visible to the user. Briefly, the software first applies an edge detection algorithm to delineate the boundary of the breast and the boundary of the pectoral muscle. Following the segmentation of the breast, an adaptive multi-classed fuzzy C-means algorithm is applied to identify and partition the mammographic breast tissue area into multiple regions, that is, clusters, of similar X-ray attenuation. These clusters are then aggregated by a support vector machine classifier to a final dense tissue area segmentation. The ratio of the segmented absolute dense area to the total breast area is then used to obtain a measure of breast percent density, PD%.
The segmented mask by LIBRA may also serve as ROIs for further radiomic analysis within CaPTk. For example, the user can focus on radiomic feature extraction within the breast or within dense tissue regions as identified by LIBRA. Moreover, radiomic feature analysis can be performed by considering either a single ROI or by applying a lattice-based approach allowing more localized feature measurements, which have been shown to leverage breast radiomics in risk prediction. More details are available as part of the breast radiomics video of CaPTk.